So today is the last uh, lesson, and we are going to talk about the last uh, way of modeling some types of biological systems, because this formalism, these logical models are not applicable to every type of biological system. So what are these logical models? First, until now we discussed most of the time models based on ordinary differential equations. And in the past week, we talked about stochastic modeling stochastic processes. So modeling biological systems, taking into account the stochasticity of the molecular processes, that is especially important when at least some components of the systems are exist in small numbers. But both approaches, either differential equation systems or um, stochastic processes, need kinetic parameters. So each process that occurs in the model, you should have a rate expression for that process, and that rate expression usually has needs parameters to be computed so that you can make simulations of your system. So the main problem in these ordinary differential equations or stochastic models is that you need a large number of parameters, and that problem is even worse when you try to model biological systems with a higher number of components or with a higher number of interactions. So more interactions, more processes, or the processes are regulated by more components, and that means that you need more parameters. And getting the right parameter values is a bottleneck in the development of differential equation systems or of stochastic models. So people have found alternative ways to, even in the absence of that detailed kinetic information, being able to model and predict the behavior of large biological systems. Um, and one of the solutions is the logical models that we are going to talk today. Um, Another solution that is, so today the logical models are applied mostly to signaling networks or to gene expression regulation networks. But if you are more interested in modeling metabolism or metabolic networks, there is also another approach that is flux balance analysis that does not try to model the dynamics of the metabolic network, so how the concentrations of each metabolite change through time, but it tries to find all the possible steady states that the metabolic network can obtain or can reach. Um, because in large metabolic networks, there is an infinite number of steady states that the network can achieve. But you can work with that using some optimization tools, and assuming that the metabolic network is regulated in a way to optimize, for example, cell growth. So, but that is, there is a book chapter that you have in the Moodle page that talks about that, but this year we are not going to give a class about that kind of flux balance analysis. And so it's not going to be part of the exam. But that's one of the alternatives that does not need kinetic parameters, but the other one is logical models. Sorry. So the The logical formalism assumes that each model variable, that is each component, can either have only two states, and that if all the model components only have two possible states, on or off, so they are active or inactive, that we can call these Boolean models. But these logical Boolean models have been extended to include some more diversity of states, but still discrete. Each node can have only a discrete set of states, and those are called qualitative logical models. And normally one of those states is the off state. So the molecule or the protein or the gene is inactive. And then there can be multiple levels of activity. So like an intermediate activity, a very high activity, but usually people don't use in this modeling approach much more than 3 or 4 levels for each node, for each component of the system. And most of the nodes are Boolean, okay? So the consequence of instead of considering, for example, for a protein, the concentration of that protein in the cell, we are only going to know if that protein is active or inactive. So the simulations lose detail. So now the simulations only allow us to follow through time which proteins or which genes get active or are inactivated a long time. But so we lose detail in the simulations, but we gain because we can build models for larger molecular networks because we don't need kinetic parameters to make those simulations. So this approach is applicable to mainly to signal transduction networks and to transcription regulatory networks. Those are the main systems that can be modeled by this approach. And this happens because for example, in signaling networks, the proteins involved in signaling pathways usually are activated or inactivated by post-transductional events like phosphorylation. So in fact, there are discrete levels of activity for many of these proteins involved in signaling. And if we are looking at gene expression, although here there are some genes may be expressed with at different rates, but for some processes, the system, what matters is if a given gene is being expressed or not so much the amount of gene expression of protein or messenger RNA that is being produced. So here it is more not so clear. But still, as we have seen, many of these, if we make models with differential equations to describe gene expression, and most of the models that we have looked with in the network motifs that Uriel studies, those involve gene expression, and all the regulatory part of the gene expression was modeled with most usually with ill functions, and if normally using a Hill coefficient higher than 1, and the higher the Hill coefficient, the more the Hill function looks like a step function. And if we have a step function, the gene expression is either on or off. So these dynamics are more likely to be well approximated by the logical formalism. So these are the two main types of biological systems that are modeled by this logical approach. Any question? No?
No.
Okay, so how can we define these models? So these models. So in the when we were talking about differential equations, the first part was converting the conceptual model or the conceptual scheme of the model of the system into the set of differential equations. In this case, we also start by the conceptual model, and it's more or less directly translated into a regulatory graph. In this regulatory graph, each node is a dependent variable of the system, usually corresponding to either a protein, a gene, a messenger RNA, or a microRNA. Um, so most of the time, the model components are these genes or proteins. And we have to define for each of the nodes how many possible states they can have. So usually they have the off state, but they can have either just one on state or multiple active states. And then the graph includes the interactions between the nodes, and these are directed interactions. So and when we have an interaction connecting node A with node B, it means that A regulates B, and in logical terms, that means that the state of B depends on the state of A. Okay, and so to complete the model definition, we don't need only the regulatory graph. We also need for each node that has incoming interactions, we have to define a logical function, and that logical function uses the states of the regulator nodes of that particular node that we are focusing on to define the possible next state for the node. So these logical functions it's like a mathematical expression involving the states of the regulator nodes and logical gates. And you can associate multiple logical operations in a single expression. And eventually you can, if a node is regulated by multiple other nodes, the regulators, so you can have quite complex logical functions. And sometimes it is easier to define the logical functions by truth tables instead of writing the logical expression. So it's an alternative way to define it. And of course, normally in these models, there are some nodes that don't have incoming interactions. And we call those nodes input nodes because they are independent variables. So normally they function as an input to the system. So the value of the input nodes, the one that you give them in the initial state of the simulation, is going to be the state for all the remaining simulation because they are not regulated. So they represent the environment where we are performing the simulations. So here is an example of a logical.
Could I ask a question?
Sure.
Do I understand correctly that so the nodes, they are defined discretely?
Yes.
Is time also defined discretely?
Yes, time is also evolves in discrete times. Also, that is when we try to compare the results of the simulations with experiments, that's possible. One of the difficulties is because these simulations occur in discrete time steps, and eventually, if all the regulatory interactions occur more or less in the same time scale, we can imagine that each time step corresponds to a given, like for example, to 1 minute or to a fixed amount of time. But if the regulatory interactions, some are more quick than others, some are gene expression regulations, others are, for example, signaling regulatory interactions. So it means that protein A will phosphorylate protein B, and that will activate protein B, for example. So that occurs in the time scale of minutes. But if it is a gene expression, meaning that there is a node A is a transcription factor that is going to bind to the promoter of gene B and stimulate the expression of gene B, that is something that occurs in around half an hour, maybe the time scale of that change.
But isn't
So,
it because I like what I understood from Uri Alon is that like the most important part why it's possible to model all these things into systems is that there is this difference in time scale, right?
yeah. So, but that normally means that that is very useful because you can either focus in modeling only the signaling or only the gene expression. So, if you are modeling the only the gene expression, what happens in the signaling part is very quick. So you can, for example, assume that all the changes in the signaling you can approximate, use, for example, a steady, quasi steady state approximation. Or if you are modeling only what happens in the signaling part of the system, you can assume that whatever is happening in the gene expression is so slow that in the time scale that you are modeling the signaling events, you may consider that the events in the gene expression are constant. So that's
But
an
aren't
advantage.
but aren't isn't signaling influenced by what is expressed?
Yeah. And actually, there are frameworks that try to model the two things iteratively. So you make a model for a small amount of time of what's happening in the signaling network, and that is feed that changes in the signaling network are feed into the gene expression model that will change slower, right? And once it changes, it will feed back into the signaling. So it's kind of an hybrid approach to model.
Okay, thank you.
But in this case, we are trying to make models of large networks, and you are right in that signal. Normally, people consider the main view is that each cell receives signs from the outside, from the environment, that normally involve a ligand activating a membrane receptor, and that will activate a cascade of events that are we usually call signaling pathways that will end up by changing gene expression in the cell. On the other hand, the gene expression will change the components of the system and may have an impact on what is the active signaling pathway that is working in the cell. So there is these two systems, the signaling and the gene expression, are interconnected, and people try to make logical models, including both types of regulatory interactions, and that's when the time step loses a bit of meaning here. But the temporal order of the changes is well modeled by this logical formalism. That's what the point I was trying to say. So
Yeah, yeah, yeah.
we don't have so there are some approaches. We are not going to talk about that today. Well, I can try to explain, but there are some approaches that try to, from the logical formalism, run the stochastic simulations that take into account the rate at which each interaction occurs. But that we'll talk about that maybe in the end. So here is an example of what a logical function looks like. For example, here for a node D, D is going to be in the on state. Normally the off state is 0, the on state is 1. So for D to be in the on state, to be equal to 1, the logical function that is after the if should be true. And so D will be activated if A and B are simultaneously active, or C is active but A is not active and B is not active. And this can be quite longer, and in this case D only depends on the values of A, B, and C, but you can have, for some nodes, you can have much more incoming interactions. And for example, in this case, we can translate this logical rule into this truth table. So you have all the values of A, B, and C at time t, and depending on the values of A, B, and C at a given time point, in the next time point, the value of D can be what it is in this column. And I say can be because the updating scheme can be different, and we'll talk about that in a moment. So, for example, if none and actually we only define the logical function for D to have the value 1. So we are assuming as D has only two states. If this expression is not true, if it is false, then D will be 0. Okay. But if we have a node that has more than two states, so a 0, a 1, and a 2, then you need to define the logical function for D to be 2 and the logical function for D to be 1. And then all the other remaining possibilities mean that D is 0. So in this example, so in this second line, if only C is active, that corresponds to this part, then D will be active also. And but if only B is active, that is not enough. If B and C is active, not enough. If A alone is active, is not enough to activate D. If A and C is active, also not enough. And then if A and B are present, no matter what is the value of C, you will get deactivation. Okay, so this is a possible logical rule. And also, as we, I think in the URL lectures, that was mentioned quite a few times. So it's quite easy to get nowadays from the high throughput technologies that people are using, it's quite easy to get the information about which nodes regulate which other nodes. So to have information about which transcription factors regulate each target gene, or the nodes that regulate each other in signaling pathways. That information is easy to get in the literature and in databases, but these logical functions are not so easy to find. So that we don't have the bottleneck of finding the kinetic parameters. It is not difficult to find which nodes regulate each other node, but it is still difficult to define these logical rules. And people sometimes what they do is they have a known set of observations, and they try to optimize the logical rules in order for the model to replicate known observations. So how do we define the interactions in logical functions? So basically, looking in the literature, that there is a lot of information available. So it is very easy to find information about the interactions. Not so easy for the logical functions, but even for the logical functions, it is easier to find information than for the kinetic parameters of the system. So that is the reason why logical models are used to model larger networks of signaling and gene expression networks. You can also use databases to find this information, and one that collects a lot of information about signaling and gene regulatory networks, at least for humans and for a mouse, not so easily for other species. This one, OmniPath, has a lot of information about these, and also tells you if those interactions, for most of those interactions in this database, it tells you if it is an activating or an inhibiting interaction. But when a node is regulated by multiple nodes, you are not sure about the logical function. And sometimes what people use is, in the absence of information, we use parsimony rules. So, for example, if a node has 2 or 3 repressors and, for example, 3 repressors and 3 activators, the majority wins. That is, if there are more active repressors than active activators, then the node will go to the off state. But if there are more active activators than active repressors, the node will turn on. So that's in the absence of information, it is easier to assume that. But sometimes we know that there are in the regulatory logic of the gene expression, for example, a lot of times there are so these parsimony rules are mostly based on OR logical gates. If we have one or more activators that will tend to activate the node. But sometimes you need to have simultaneously the presence of two activators to have the consequence of activating the target. So that information is more difficult to obtain, but sometimes it is described. So you start by using these parsimony rules, and then you try to fit experimental data. So you know that after some knockout experiments or overexpression experiments, you follow gene expression or phosphorylation state of a set of proteins a long time, and you check what is the time evolution of those activations or inhibitions and of several nodes in your model, and you try to check which logical rules allows you to have to simulate those temporal evolutions of activations, inhibitions of the components. And actually, there is more in the literature. We find more frequently these kind of gene expression studies after knockouts or after overexpression experiments than we find quantitatively quantitative time courses where you follow the concentration of the components through time. So it is easier to find experimental data to try to adjust these logical models. And also there is these logical models and Boolean networks are frequently used, for example, in people developing electronic circuits, right? Because electronic circuits are basically a network of electronic components that implement these logical gates and not and or and I only use the and, the or, and the not because with these three basic logical functions you can get other logical functions. For example, there is a XOR logical gate, so that is can be composed of ands or and not. Where was I? So okay, so this modeling of Boolean networks is quite used in electronic engineering, mainly in digital electronics. In that field, for example, if they want to design a circuit that is able to perform a given operation, so in face of a given stimulus input, it will produce a given response eventually with a given temporal order. There are techniques called model checking that try to do that even without making the full simulation of the model trajectories. So they test if a given structure of the model is able to reproduce a given trajectory. And that is a powerful approach, although it was originally developed mainly for the design of electric integrated electronic circuits. Okay, questions? So the simulations here are quite different from what we saw with either systems of differential equations or with stochastic systems. So as we said before, the simulations are performed in discrete time steps, and the state of each component at step n is defined by the state of its regulators in the previous time step. But there are different strategies to make the update of the variables at each time step. So the most basic one is the synchronous update, where you compute the values of all the logical functions for every node, and all the nodes that should change according to their logical functions, they can change simultaneously in the next time step. So that's the synchronous update. But you can also use a synchronous update. That means that in each time step, only one of the variables that tends to change according to the logical functions only one changes at a time. Why people do this? Because as we are not sure which of the transitions occurs first, when we model all in the synchronous update, all changes occur at the same time. But if in reality one of the changes occurs first, that may have an impact on the other logical functions. So if each time one node changes from on to off or from off to on, that may have an impact on other logical functions. So one transition that should happen before, maybe it's not necessary anymore. Or after one transition occurs, then you have much more transitions that should occur. So in the asynchronous method, you simulate all those possible transitions. And trying to solve that problem that we already know that some of the processes that are being modeled are occurring in a shorter time scale than others. It is possible to define asynchronous updates with priority classes. That means that if I have multiple possible transitions to occur from between two consecutive time steps, I will give priority to the ones that I know that occur at a smaller time scale, so that are quicker. And that process can be done so there are different flavors of these asynchronous update with priority classes. So if all the transitions in the quicker priority class will occur synchronously, or you can pick just one of the transitions in the high priority class. So you can still have variations in this update scheme. So in the synchronous update, all the nodes change, and that means that if you start the model, the simulation at some state of the system, only one trajectory is possible. Okay, but in the asynchronous scheme, as only one node is updated in each time step, if you are following just one trajectory, then you have to choose randomly which of the updates of the nodes updates its value, or then you are modeling in parallel multiple possible trajectories, but from a given initial state, you have multiple possible trajectories. Not all of them eventually are equally likely to occur because some processes may be inherently more slow or quicker than others. But in the asynchronous update, you get all the theoretically possible trajectories of the system. And in the asynchronous with priority classes update, then you try to approach the asynchronous update to a more likely to occur transition in reality. But you can notice that in the asynchronous with priority classes, it's like we need some kind of parameter for each interaction. We need to know if it is quicker or slower. So we start having the need for some kind of parameter, and that is also a reason why you don't see a lot of published models with the asynchronous update with priority classes. Most of the times you see models either with asynchronous or with the asynchronous update. Because the priority classes already imply the definition of parameters, and the need for parameters was what led people to develop this approach. So to have a way to model the systems without needing so many parameters. And one way to do the simulation without having to randomly choose the next state, so which node is going to be updated, is to represent all the possible trajectories, and that is what is done when we build a state transition graph. So one way to represent the simulations in these logical models is to build a state transition graph where each node now is not a variable, it represents a possible state of the system. Okay, so each node is identified by the values of every variable in the system. And directed interactions between two nodes in this state transition graph represent a possible transition between two states in between two consecutive time steps. So if you find the path along the state transition graph, it's actually a possible trajectory of the system. And the state transition graphs are graphs, so that you can find communities with them, you can find shortest paths, you can find components. And one thing that is useful to find in state transition graphs is that to identify the attractors of the system and the respective basins of attraction. So you can now, it's like this state transition graph is equivalent to the phase space in the differential equation. So the phase space contained all the possible states of the system. So the coordinates of the state space, sorry. The coordinates of the state space represented all the possible, a given possible state of the system. Now, each possible state of the system is one node in the state transition graph, and a path in this state transition graph is a trajectory of the system. Attractors are one state of the system or a set of states of the system that, if the trajectories go into that set, they will never get out of that set. So what kind of attractors can you have? You can have fixed points when you have one node in the state transition graph, so one unique state that has only incoming arrows. So you can, trajectories can go into that state, but they will not go out of that state. Why? Because in that state, all the logical functions will agree with the current state of the variable. So no node needs to change. But you can also have a different type of attractor that we call cycles, that are sets of nodes that form a strongly connected component. We'll see in a minute what is a strongly connected component. But you can imagine that you have a set of nodes that are interconnected between them. So you can have paths occurring within those nodes. But outside of that set, there are no outgoing arrows, so only ingoing arrows. So trajectories can reach that cycle, but then they keep cycling between those states forever. So that's another kind of attractor, called cycle. But strongly connected components, and here is the strongly connected components are defined as directed subgraphs, where every node can be reached from any other node in that subgraph through a directed path. But strongly connected components, to be an attractor, they need to be, their interactions with the outside, with other states, are only through incoming arrows. If they have outgoing arrows, it means that a trajectory can go into the strongly connected component, can go around visiting several states in that strongly connected component, but at some point it can go out. So it's no longer an attractor. So you can have strongly connected components that are not necessarily attractors. And the system trajectory can be retained within that strongly connected component for a long time. But if it has some probability to escape that strongly connected component, then it is not a true attractor. But it will have an influence in the dynamics of the system. So here's a small example. For example, here we have a system component. This in the left is the regulatory graph. So you have each G0, G1, and G2 are the nodes, the components of the system, are proteins. And for example, G0 activates G1, G1 inactivates G2, and G0 activates G2, but G2 inactivates G0. So you have to define, for the logical function for G1 is simple. If G0 is active, G1 will become active. If it is off, it will become off. For G2 is also simple. No, for G2, you have two incoming interactions. So there should be, I don't remember what was the logical rule that was implemented. But here, for example, we'll see that in a moment. So, but there should be some logical rule combining the effect of G0 and G1. And G0 also has a simple logical rule because it is only influenced by the state of G2. So this is the complete state transition graph. So it represents all the graphs. In this case, it is simple to observe because the model only has three variables, so three dependent variables. So you only have the values for the three nodes. And if you start, for example, in the 0, 0, 0, so all the nodes are off, there's only one possible transition, that is going to the activation of G0, because if all the nodes are off, then G1 will keep off because G0 is off. G2 also keeps maybe off, but if G0, if it is not, yeah, because G0 is off, it will not activate G2. So G2 is not repressed by G1, but is also not activated by G0. So it will keep being 0, and G0, as it is not activated by G2, it will become activated. Once you have in the 100 state, you can migrate to two different states, either to the 110. So G0 is 1, G1 is 1, and G2 is 0. And this is a stable state, is a fixed point, because once you reach this node, you are not able to get out of the system, because in this state, all the logical rules predict that all the nodes should maintain their value. So it's just a fixed point. But if you go to 101, then you can have again two possible transitions, to 001 and to 111. And in fact, in this case, you can, for example, you have here a possible cycle. You have 101 converting to 001, 000, 100, going back. So the system can be forever repeating this cycle. And this, in fact, is a strongly connected component, okay? But it is not an attractor because it has escape routes. So at some point, it will, either in this point or in this point, it can migrate to 111, or it can migrate to 110, okay? So you have, in this state transition graph, all the possible time trajectories of the system. Questions?
So the 110 is when there's G0, there's G1, and there's no G2, right?
Yes.
So if there are no arrows going out, probably for the G2, the logical expression or function, it's probably that there has to be G0 and
not
no G1, because
exactly.
so that's why it doesn't leave, because if it was if there was just some G0 and some G1, it could be possible, but maybe
nice.
not.
You are ready to go on and making logical models. So you already have a logical how do you say that, thinking.
Yeah.
So, but that's it. So okay. That you are completely right. So in this case, it is a very simple model, but you can apply this to larger models. And in these systems, it's not so interesting to follow exactly the dynamics. So it is more interesting to follow which are the possible stable attractors and what do they represent, because this is if this is a gene expression, it means that some cells will be trapped in a setting where they will express G0, G1, but not G2, and that may have a functional consequence. And in practice, many of these models are used to study cell differentiation. So a cell starts being a stem cell, and it can differentiate into different types. and each of those different types can be a different attractor in this state transition graph. And you can study not only the existence, the number of attractors, but also what is their basin of attraction. In this case, wherever you start, so you just have one stable attractor, and wherever you start, you for sure, after a given amount of time, you will end up in the stable state. So the reachability, the probability that the trajectory will end up in this state after a long time, it is 100%. So that, but if you have multiple stable steady states, you can study how likely it is if you start at a given node, at a given state, how likely it is that you end up in one stable state or in the other. And that is what is called the reachability of the different attractors. And of course, you can define the basin of attraction. In this case, the basin of attraction is the whole steady state transition graph, because wherever you start, you will eventually end up in this stable fixed point. Yeah, if you have multiple, you may have different regions of the state transition graph that are the different basins of attraction. Okay, and that may also help you to decide. The larger the size of the basin of attraction, the larger the reachability of the corresponding attractor, because it's more likely that some trajectory will step into that basin of attraction, and once in that basin of attraction, it will converge to that stable steady graph. And in this case, while in the continuous deterministic models with differential equations, all the points in the state space or in the phase space are within a basin of attraction. In this case, you can have, you could have a set of states where you could either migrate to one steady state or to a different one. So there are some nodes that eventually are not part of any basin of attraction. So that's another difference in these kind of models. Any question? No. So the questions that we can try to answer with these models is, for example, if this network of signaling or of gene expression regulation is responsible for the differentiation of the cell state. You can find the attractors, and those attractors will correspond to a possible cellular state or cellular differentiated state. You can estimate the reachability, that is the probability with which an attractor is reached by a system. And remember that you have many models, not the example that we looked at. Here, there is no input node, because every node, every one of the three nodes has incoming interactions, but even in some systems, you have nodes that are not regulated by the system, are independent variables. And in those cases, you can study, depending on the input node values, what are the most likely attractors to be reached. So that's one of the ideas of using these models, is trying to understand which are the environments that lead systems to different cell states or to different differentiated states. You can also perform stochastic simulations on the state transition graph that correspond to random walks on the state transition graph. And that is the case where it is possible to make, instead of working with the continuous time step, you can do these stochastic simulations on the state transition graph, taking into account the time it takes to change from one state to the other. And that time is a function of what is the node that is changing its value. So you can make this random walk, but you can estimate randomly, looking at what node changed its value, so what kind of interaction originated that change. You can estimate from an exponential distribution the time that transition took to occur. And then you can perform multiple of these stochastic simulations during time, and then follow through time and considering different possible time steps for each transition. And if when you average those multiple stochastic simulations, you can transform that discrete value simulation into a continuous scale that will give you, if all the nodes are Boolean, that will give you at each time point what's the probability that that node is active. Okay, and actually people have done this approach, so performing stochastic simulations in the state transition graph, and then compared the time evolution of the probability of a node being active, and compared that with continuous data about the activity of the nodes. And they were able to predict the time evolution of those nodes, basically because it's like each individual stochastic simulation is like what is happening in one cell, but in most experiments you are observing changes in populations of cells, so they average out. So these average of multiple stochastic simulations may be correlated or compared with the quantitative evaluation of the activity of the nodes a long time. Yeah, and actually the one of the fields of activity where these models are more applied is in studying cell type differentiation, also in studying cell cycle regulation. Also, there are a lot of models, and I think this is the last slide that I have. Questions? No. Now I'm going to show you that you can easily start to use this kind of modeling. There is a free where is it? Extend session. Okay. There is a free software. No, this is the course. Sorry, there is a free software called GeneSim. That you can easily set up one of these models. You have yeah, this link in the Moodle page will lead you to a paper that actually is a tutorial on how to use GeneSim to build a model like this one. And will guide you through how to implement the model. And GeneSim, I have here the GeneSim. This is the home page of GeneSim. You can download. It's a Java-based application, and actually I have it open here. Here is a model of T-cell differentiation. So this model is a simple one that tries to explain how T cells differentiate either CD4 T cells differentiate either to Th1 or Th2 phenotypes. So there are some nodes here that are the environment, so they are not regulated, and depending on the presence or absence of some of these cytokines, the cells will tend to differentiate either into the Th1 T helper 1 phenotype or to Th2. So, but this only explains the simulation, the differentiation into two cell types. But let me check if I file open. So this one is a much more complex model that also studies the differentiation of T cells, CD4 positive T cells, but now into not only into Th1 and Th2, but into I think around 9 cell types. So Th1, Th2, TH9, TH17, Treg. So these are the T regulatory cells. TH9, I said, gamma delta T cells. And that is explained through so there are a lot of input nodes. So a lot of these variables define the environment, and depending on the value of the input nodes, the different populations of T cells are more or less likely to be reached. So each cell type corresponds to an attractor, to a steady state in this model. And you can use this tool, the GeneSim, to define the model, to build this regulatory graph, and to build so maybe in this smaller one, if I choose a node, I have here the possible regulators. So EL4 is regulated by EL4 receptor, is regulated by EL4, and it is inactivated by SOX1. And the EL4 receptor will be active if EL4 is active and SOX1 is not active. But you can also define this as not as, in this case, it is defined not as a formula, but you can write here the logical formula. Alternatively, you can define here what are the set of active interactions that will lead to a given value. So there are different ways to perform to define the model. And you can also find the stable steady states. So find which possible steady states are achievable with this model and run a simulation that in practice is building the state transition graph. Okay, so that's what it is. And if you go through that paper, you can install GeneSim, and if you go through that paper, you can see how these analyses are done. And if you actually, this GeneSim is quite nice to visually develop the model, but if you want to make more large-scale simulations, the community of logical modeling has developed this Colomoto consortium that is based in Python. So it's a library of Python algorithms that and software tools that can be used to simulate, to analyze this kind of logical models. Okay, so that's it. Questions? No questions?
So you finished the class, the theoretical class?
Yes.
Ui, okay. Congratulations to us, it's ended. Okay, but I actually have a class about the TP, but I'll let my colleagues, in case they have doubts, to expose them first.
Yeah. Any questions about these logical models? So if we have an extra TP, we could go through that paper and follow the use of GeneSim. But I think that is also something that you can do on your own if you are interested in trying to apply. So these logical models are also, so in the beginning, we start by looking at interaction networks, and these logical models are, it's a more straightforward way to go from an interaction network to a dynamical model. Although with some limitations, because it does not allow you to simulate the full dynamics of the system. It is a discretized dynamics. Okay, and also these logical models are not covered nor in the Brian Ingles book, nor in the Uri Alon's book, but I think in Moodle I have a review paper that covers what we are. Yeah, there is a review paper in the lecture materials that about logical models that you can go through to study this part. If you are curious, there is also a chapter about the metabolic flux balance analysis, but it will not be covered in the exam this year. Okay, I will stop the recording.